Welcome back, everyone. Today, we're going microscopic. We're going to dive into the world of mammalian cell culture.
It might sound niche, but trust me, it's more relevant than you think. We're talking advancements in medicine, research, cosmetics, even food production.
Wow, okay, I'm already hooked. But before we get ahead of ourselves, can we get a little cell culture 101? Like, what are we actually talking about here?
At its core, it's all about giving cells the right conditions to thrive outside their natural environment. Think of it like creating a perfect little world just for them.
Creating a world. Okay, I'm picturing tiny, happy cells living their best lives. But how do we even begin to design that perfect world? What are the essentials?
Well, first off, we need to get the temperature just right.
Okay, that makes sense, keeping them comfy. But I imagine it's more than just a temperature-controlled room.
Oh, definitely. Think of it like making a recipe. They need the right ingredients to thrive, and that's where the culture medium comes in.
Culture medium, like their food.
Exactly. It's a special liquid packed with all the good stuff. Amino acids, vitamins, salts, glucose, you name it. It's like their all-inclusive buffet.
Sounds pretty fancy. But speaking of keeping things pristine, I imagine contamination is a major concern.
You're absolutely right. Contamination is a huge deal. Even the tiniest speck of bacteria or fungi can wipe out an entire culture.
So how do you combat that?
Sterility.
Yeah.
Everything has to be perfectly clean, the workspace, the tools, every single thing. We work in these special biosafety cabinets and use strict sterile techniques.
It's like operating on a microscopic level, right?
Yeah.
Every move has to be precise and sterile.
Exactly. Now, beyond the environment, we need to consider the type of cells we're working with. We've got two main types, adherent cells and suspension cells.
Ooh, okay. Different types, different needs, I'm guessing. What makes them unique?
Adherent cells are, well, they like to stick to surfaces, like the bottom of a flask, for instance. Think of them as the home bodies of the cell world. They need their own little spot.
So they like to settle down. Makes sense. And what about those suspension cells?
Suspension cells are more like free spirits. They happily float around in the culture medium, unattached to anything.
Ah, so they're the adventurers roaming free.
Exactly. And this difference in their lifestyle means we need to care for them differently too. How we handle them, feed them, even how we move them depends on whether they're adherent or suspension.
Move them. Wait, what?
Yeah. As cells grow and multiply, their little world can get pretty crowded. So we transfer them to a new, larger space with fresh medium. It's a process called passaging.
Passaging. Got it. Like moving them to a bigger house when they need more room.
Yeah.
Is that different for the two types of cells?
Definitely. With adherent cells, we first have to detach them from the surface they're clinging to, then dilute them and move them. Suspension cells are much easier. Just transfer a portion of the cell-filled medium and dilute it in a new flask.
Got it. So it's like carefully picking up those adherent cells from their little homes while the suspension cells are already packed and ready to go.
Yeah.
But whether they're home bodies or adventurers, I imagine gentle handling is key throughout this whole process, right?
You bet. We don't want to stress them out or damage these delicate cells during the move.
Makes sense. Okay, so we've got them settled in their new digs. What else do we need to do to keep these cells happy and thriving?
Well, just like any living thing, they need regular care and attention.
Like us, I suppose.
Absolutely. So we need to keep an eye on their environment, the temperature, the pH, and make sure they get regular changes of fresh medium.
Ah, so like refreshing their food and water and keeping their space clean. What are some signs that maybe things aren't going so well? Like, how can we tell if our cells aren't feeling their best?
There are a few things to watch out for. If the medium starts to turn yellow or cloudy, that could indicate there's a buildup of waste products.
So it's like their little world getting polluted.
Exactly. Another thing we might see is a change in their growth rate. If they slow down or change shape under the microscope, that could be a sign that something's off.
Fascinating how observing these tiny changes can tell us so much about their health and well-being. It's like they're communicating with us in their own way.
I like that way of thinking about it.
Okay, so we're learning to speak cell. But what happens if, despite our best efforts, things still go wrong?
Well, sometimes we need to hit the pause button on these cells, and that's where cryopreservation comes in.
Cryopreservation. Sounds intense. What is that exactly?
Basically, we freeze the cells to preserve them for later use.
Wait, freeze them? Wouldn't that, I don't know, damage them? Cells are pretty delicate, right?
You'd think so. But we use a special solution called a cryoprotectant, usually DMSO, to protect them during freezing.
Cryoprotectant, DMSO. Okay, so it's like giving them a little shield against the cold.
Exactly. It prevents ice crystals from forming, which would be really bad news for the cells. Then we slowly cool them down to super low temperatures, like negative 80 degrees Celsius or even lower in liquid nitrogen.
Liquid nitrogen. Wow, that's getting pretty serious. So how long can they stay frozen like this?
Theoretically, forever. It's like putting their biological clock on hold.
That's pretty amazing. And then when you need them again, you just, unfreeze them?
Yeah, we thaw them out quickly, wash away the cryoprotectant, and they're good to go.
It's incredible how resilient these cells are. But we've talked a lot about ideal conditions and keeping things clean. But what about when things go wrong? Because, let's face it, sometimes stuff happens.
Oh, absolutely. Even with the best care, challenges can pop up.
Like what? Give me the worst case scenario.
Well, contamination is always a concern. One tiny bacterium or fungus can take over an entire culture.
Oh, man. It's like a microscopic invasion, ruining our perfectly crafted cellular world. How do you even prevent that?
It all comes down to aseptic technique. Everything has to be sterile. The workspace, the tools, everything we use.
So it's like performing surgery, but on a miniature scale, with every move requiring absolute precision and cleanliness. Intense.
You got it. But contamination isn't the only thing that can go wrong. Sometimes, despite our best efforts, the cells just aren't happy.
Unhappy cells. Okay, how do we know when our cells are having a bad day? What are the signs?
They might grow more slowly or change shape under the microscope.
So it's like they're sending us distress signals, huh?
Exactly. And when that happens, it's time to put on our detective hats and figure out what's going on.
Cell detectives. I love it. So what's the first clue you look for?
We examine the culture conditions, the media, look for any signs of, anything unusual.
So it's a bit of a puzzle then, trying to figure out what's stressing them out and how to get them back on track.
That's a great way to put it. It's a combination of science, observation, and a little bit of intuition too.
Wow, it sounds like being a cell culture expert requires not only scientific knowledge, but also a keen eye for detail and a bit of a sixth sense for what those cells are trying to tell you.
It definitely helps. And all that effort is worth it because cell culture allows us to do some pretty incredible things.
Like what kind of amazing things have we learned about human biology thanks to cell culture?
Well, cell culture has revolutionized our understanding of, practically everything. How cells communicate, how diseases develop. It's given us a window into the microscopic workings of the human body.
It's amazing how much we can learn from these tiny cells in a dish. Like, can you give me a specific example?
Sure. Take cancer research, for example. Cell culture allows us to study how cancer cells grow, how they spread, and even test different drugs to see what might stop them.
So by isolating those cancer cells, you can learn how to fight them more effectively. That's incredible.
Exactly. And it's not limited to cancer either. Cell culture has helped us understand a whole range of diseases, from Alzheimer's to HIV.
Wow, so these tiny cells hold the key to unlocking some really big mysteries about human health.
Absolutely. And it's not just about understanding what goes wrong. Cell culture is also being used to develop new therapies.
Really? Like what? I'm so intrigued.
Well, scientists are using cell culture to grow skin grafts for burn victims, for instance, and they're even developing cell-based therapies for things like Parkinson's disease.
So it's not just about studying what's broken, it's about using these cells to repair and regenerate tissue.
Exactly. Cell culture is opening up a whole new world of possibilities in regenerative medicine. It's pretty exciting stuff.
It's mind-blowing to think about the potential. From understanding diseases to actually developing treatments, it seems like cell culture is at the forefront of so much medical innovation.
It really is. And the most exciting part, we're just getting started. As our techniques and technologies keep advancing, who knows what incredible discoveries and applications we'll see in the future.
It's inspiring to think about. These tiny cells that most people don't even think about have the potential to transform human health.
I couldn't agree more. It's a testament to the ingenuity of scientists and, just the sheer power of biology.
Well, I think we've covered a lot of ground today, from the meticulous care it takes to keep these cells happy to the incredible potential they hold for scientific advancement and even medical breakthroughs.
Yeah, I think so too. Hopefully this gives our listeners a little glimpse into the amazing world of mammalian cell culture.
It's a field that most people don't even know exists, but it's clearly making a huge impact in so many ways.
You said it. And who knows, maybe one of our listeners will be inspired to join the ranks of scientists pushing the boundaries of what's possible with these incredible cells.
That would be fantastic. It's a field that's just teeming with possibilities.
It certainly is. So, to everyone listening, if you're curious about the world at a microscopic level, cell culture is definitely worth checking out.
Absolutely. And with that, we'll wrap up this deep dive into the fascinating world of mammalian cell culture. Thanks for joining us.
It's been a pleasure sharing these insights with you all.
And until next time, stay curious.